O God of creation, direct our
Amen.
Amen.
Tired of constantly juggling tabs while working with web apps on your...
So sorry, my name is Olani Christiana. I'll be your moderator for this evening. And today we'll be taking the topic, traditional and emerging anti-infectives. Traditional and emerging anti-infectives. And it will be taken by Dr. Shinaya Onodugo. But before we proceed on this, I'd like to recognize the presence of everybody here. Thank you for taking time out to be here. Thank you for making it a day with us. We'd like to recognize the National IDPaN National ESCO here present. I don't know if you can recognize any here right now, but the IDPaN National ESCO would like to say thank you for this opportunity to make this presentation. They would like to say thank you to the desk officers of the Infectious Disease Pharmacists Association of Nigeria for putting up this program. And all our senior colleagues here on the call, I'd like to say thank you for being part of us this evening. We appreciate you and we welcome you. The fellows of the Pharmaceutical Society of Nigeria, fellows of Postgraduate College of Pharmacists, thank you for being here today. Right now, I'd like to call on Dr. Issa for a brief opening remark before we go on to the program, the presentation. Thank you, Dr. Issa. Please take the opening remarks.
Good evening, everyone. I hope you can hear me loud and clear. So, good
yes,
evening.
we can hear you.
You are all welcome to this our presentation tonight. The Infectious Disease Pharmacists Group, like we all know, is a specialty group of the Clinical Pharmacists Association of Nigeria. The essence of bringing up this group is that a time has come that as pharmacists, we should also try and go beyond just being a generalist and looking for a place that we could be called experts. Luckily, just about the time that CPAN initiated this idea of a specialty group, our college, the West African Postgraduate College of Pharmacists, also started bringing the issue of specialization in training. I think for the past 3 to 4 years now, we've been having graduates that have 1 or 2 areas of specialization. In our own case, infectious diseases, what we are interested in is the use of antimicrobial agents in rational manners, in a way that we are sure that as pharmacists, as we safeguard all drugs, we know that antibiotics have special places. And at any time that we want to talk about safeguarding drugs, antibiotics come first. Because while an error in one drug, an analgesic, for instance, may have just an effect at that particular person to that individual, that of antimicrobial agents may have effect on the future and against other people. So we say that let us go ahead and start talking about how to specialize in infectious disease, how to use drugs. But in today's edition, what we want to do is that we've been talking about antimicrobial agents. We've talked about different topics. We brought presenters from different parts of the world, from the US. We have had two presenters from the US, one being a fellow of the American College of Clinical Pharmacists. But tonight, we said that let us come down to talk about the antibiotics themselves. What are the traditional ones that we know? What are the new ones that we also ought to know? And what should be the position that will give them in the selection of all these drugs? These are the things that we want to learn tonight. And we have in our midst somebody that is well grounded in it. I'm sure whenever it's time for the reading her citation, justice will be done to that. Thank you very much.
Much, Dr. Issa, for that brief remark. We appreciate you. Before we go on, I would like to remind us about a few housekeeping rules that we should try and keep our mic muted, please. If you're not talking, especially during the presentation, please keep your mic muted so that we can all enjoy the presentation. And to mark attendance, please put your names. Your names.
Now, what's the basis of the story now?
Please mute your mic. Emmanuel Ede, please mute your mic. Thank you. So please, to mark attendance, let's put our names. If you're a member of Infectious Diseases Pharmacists Association of Nigeria, please put, and probably your CPAN number to mark the attendance. So I'll go ahead and take the speaker's citation. The speaker, Dr. Mrs. Chineye Onodugo. She had a BPharm, Master of Pharmacy, PhD in Clinical Pharmacy from the University of Nigeria. Then she had a PharmD from UniBen. She's a deputy director at the University of Nigeria Teaching Hospital. She's in charge of the accident and emergency, so also the pediatric intensive care unit at UNTH. She's the secretary of Faculty of Clinical Pharmacy of the West African Postgraduate College of Pharmacists. She's also a state coordinator for Enugu State. She has won different grants related to antimicrobial stewardship. And this has taken her to make her presentation globally in different places. She's very passionate about antimicrobial stewardship. She's also a preceptor to undergraduates and postgraduate students of the University of Nigeria, and also to residents of West African Postgraduate College of Pharmacists. She's knowledgeable, she's passionate, and she's very astute when it comes to antimicrobial stewardship. So sit tight tonight and let us enjoy this presentation as I call on Dr. Mrs. Chineye Onodugo to present on traditional and emerging anti-infectives this evening. Please come on. Thank you. You're welcome. Dr. Mrs. Onodugo.
Trying to track her on the group. I can't see her name, but she's good to use another device. Let me see the name she's using.
So while we are waiting for her to come on, I think it was last week we celebrated the World Antimicrobial Resistance Awareness Week. So different programs has been on to increase awareness among stakeholders, so that among the public, among stakeholders, so that everybody will be in the know and fight antimicrobial resistance together. We have the traditional ones that we have known. We have the emerging one that maybe most of us are not aware of. So that is our expectation tonight, that as an expert, she will take us through and will be on top of knowledge, abreast of what is happening around the world concerning antimicrobials, their effects, and the new ones that I just imagined, and how to protect it so that we can combat resistance, especially to the new ones. While we are still protecting the old ones, the traditional ones that we know, we can also work on getting to know the new ones and work on preventing resistance to them. Have you been able to track us?
She has received the link. She's joining now.
All right. Please, just a few moments, you'll soon be part of us.
The phone decided to have an issue on the day that we needed it the most. Just a few things before she joins. After tonight's meeting, the infectious disease pharmacist group will now have 2 more sessions before we close for the year. Next week, we're going to have an overview of all our clinical presentations and journal club presentations that we have done for the year. You know, we've had since the IDPaN conference ended, we've had up to 6 to 7 different case studies being presented in our WhatsApp group. We have some case studies that are simulated just from textbook, and also have those ones that are directly from the bedside of the patient, where colleagues from the hospital pharmacy and even from the community pharmacy bring those cases and we discuss them. We've also had an infectious disease debate where we had 2 people from the hospital and 2 from the community pharmacy talk about the roles that each of them will have to play in preserving our antimicrobial agents. But beyond that, one of the things that we'll be doing is the journal club, where we bring out publications about antimicrobial agents and we do critical appraisal of them. Dr. Joda has been too engaged in that and has been guiding us to producing experts who can bring out evidence from publications without being biased. That's what we do in the journal club. And I think we've had 6 so far. From the beginning to now, we've had 6 different articles that we have appraised. Members in groups, sometimes together, we'll talk about different publications from local and even international presentations, where we try to see if there are flaws and limitations in their designs. We also look for the strengths of those publications. And at the end, we take a decision about the conclusion of those presentations. I think we've had 6, and all of them, by next week, we're going to have a meeting where Dr. Onodugo will come, sorry, Dr. Joda will come and tell us about what we've done so far and what will be the way forward. Okay, Dr. Onodugo is online now, but trying to join with her device. Dr. Laine, can you see her now? I think I see her online.
I can't see her yet.
She's online now. I've asked her to.
She's online. Okay. So Dr. Onodugo, you are welcome, ma. Hello, can you unmute and speak, please? Hello, ma, Dr. Onodugo. Hello, ma.
Can you hear me?
Yes, we can hear you now. You're welcome on board, ma.
Hello.
Hello, ma. We can hear you, ma. Can you hear me?
Can you hear me?
Okay, you're welcome, ma. So you have about 45 minutes for the presentation, ma. So you can share your screen.
Are you sure she can hear you? I'm not sure she can hear you yet.
Hello, can anyone hear me?
We can hear you. We can hear you, ma.
Hello.
Yes, we can hear you.
Okay. I can't hear anything.
We can hear you. You can share your
Good
screen.
evening.
Good evening. Welcome.
So, all of the evening, and I was sincere. Hold on.
I don't know. It's like the internet is not very strong. It's breaking here.
It's breaking so much.
Hello, ma. Like she's no longer online. Okay, she's still with us. Hello, Dr. Nodugo. Where is it now? Hello, ma. Can you share your screen, ma?
I don't think she can hear us because, okay.
I would like to suggest. Hello, can moderator hear me?
Yes, I can.
I would like to suggest instead of just waiting indefinitely, maybe we can be using the time productively. Maybe we can just ask some people to share with us some of the experiences that they had or some of the activities that they had in marking the antimicrobial awareness week, or some of the things that can be suggested that we could do maybe as CPAN or as IDPaN to um, celebrate antimicrobial awareness week in our various practice areas or something, so that we can use the time productively rather than just waiting until she can communicate. There's nothing we really can do. So instead of just waiting indefinitely, I think we can begin to utilize the time for something. Yeah, my suggestion anyway.
Thank you very much, ma. That's a very good one. So we like to encourage comments or
Hello.
experience. Yes, ma, we can hear you.
Oh, finally. Oh, please,
Oh,
my
Yes,
thank
it's
God.
better now. It's better now.
Oh dear, ma. My apologies, please, everyone. Let
Okay.
me, maybe I can start sharing my screen.
Yes, please. We're so sorry for the itch.
No, it's not your fault. And I don't
No, no
know. I've
problem, ma.
never really had, I
I
always
was
have a nice time when I have to do online lectures. I don't know what happened today. But it's okay.
all
We
right, ma. So
thank God
you're
for everything.
welcome,
Can
ma.
everyone see my screen?
We can see your screen.
That's fine.
Okay.
You can
All
put
right.
it
So
on the slideshow, ma.
it's on show.
Okay, okay. So you have about 40 to 45 minutes for the presentation, ma.
It's okay. No problem. We'll make it work.
All right.
So good evening, everyone. Today we're looking at traditional and emerging antimicrobial agents. This is kind of a recap of a lecture we've had before. So I decided to just flip through some of the old things and then we'll talk about a few emerging antimicrobial agents. I must raise a caveat here that I in no way was able to exhaust all the emerging antimicrobial agents because there's so many of them on several phases of clinical trials. And we're hoping that they all pull through because, like we all know, our antimicrobial pipeline is drying up. So anything that we can get, we'll be glad to. So just able to touch a number of them that have passed through clinical trials and have started being in use. Even though that because most of them are 4, 5, 6 years old, we don't have a lot of data yet on their post-marketing surveillance. So we know what chemotherapy is, and we know what an antibiotic is. We know the history of antimicrobials and who discovered it for the first time. And I always tell people that the same person who discovered antimicrobials, discovered penicillin in particular, was also the first person to warn against the possible effect of antimicrobial resistance. And his warning, which was made as far back as 1945, has come to stare us in the face. We know that generally we have the gram-negative and the gram-positive. And I always like to refer to this because sometimes we cannot talk about antimicrobials without talking about whether they're active against the gram-positive or the gram-negative. Usually, the gram-positive are usually easier to get to. They have an outer layer of peptidoglycan, whereas the gram-negative has an outer membrane of lipopolysaccharide covering that peptidoglycan. So it's usually a little more difficult. And that's why you would find that gram-negative, multi-drug resistant gram-negative infections are quite difficult to handle. We also remember that most organisms are either spheres or rods and so on. And these are just reminders. We know that the staphylococci tend to form clusters, while the streptococci or the enterococci form pairs or chains. And this helps you if you're going to do microscopy as a preliminary result while you're waiting for the 48 to 72-hour culture to come out. So if under the microscope, they're able to identify that it's clusters or rods or chains, then you're beginning to decipher what actually the organism that is the offending organism in the infection you're trying to manage is. So I'm going to skip all this because we've lost time, but I'll share the slides. I made them a little bit wordy so that you can actually study it on your own and it will make sense to you. In general, we remember that some antimicrobials have very similar oral to IV bioavailability. And because of that, they are very, very good options when you must give even very sick patients antibiotics by mouth. So because the quinolones, the metronidazole, doxycycline, and linezolid have very similar oral to IV. So except if it's severely life-threatening or the patient's GIT is not functional, you could actually do the orals and get a very similar outcome. Remember that the quinolones and the tetracyclines, the absorption is impaired by minerals. And then protein binding, abscesses, and I must keep emphasizing abscesses. It's difficult to have effective antimicrobial therapy in the presence of an abscess because the antimicrobials do not get distributed to abscesses. So usually there must be the incision and drainage for you to get optimum effects. Of course, several antimicrobials are excreted unchanged, and you must adjust the dose in renal impairment. Being excreted and changed, if you remember, means that it does not undergo biotransformation. So it's toxic to the renal tissues. Remember the concentration-dependent antimicrobials and the time-dependent antimicrobials, and that factor, how it impacts on how the drugs are dosed. So the time-dependent, you have the beta-lactams, the macrolides, and so on. And the important thing is that they need to have a long time above MIC. And it is the time that they are able to sustain levels above MIC that determines how frequently the drug is dosed. All right, so the optimal responses occur when the drug remains above MIC for up to 50% or more of the dosing interval. And so usually you see the time-dependent antibiotics are normally dosed more frequently in the day than the concentration dependent. And then you have the post-antibiotic effect, which is very minimal in beta-lactams. That means after the patient has finished the antimicrobial, you're still having persistent suppression of bacterial growth. It's minimal in beta-lactams. You see it a lot more in the glycopeptides, that's vancomycin and linezolid. I've talked about the concentration dependent and the time dependent. So remember the beta-lactam antibiotics where you have your penicillin, your cephalosporins, your carbapenems, the monobactams. And remember that their major issue is anaphylaxis or hypersensitivity. And then in very high doses, you have seizures. Please recall that most beta-lactam antibiotics will require renal adjustment when the patient's renal function is compromised. And they usually have just the same mechanism of action which is inhibition of the penicillin binding proteins in the cell wall and leading to autolysis and cell death. And please recall that most of them have little or no activity against the atypicals. That's why anytime you have atypical infection suspected or confirmed, you would have to go to the macrolides. Okay, I'm just brushing through. So the beta-lactams are bacteriostatic and bactericidal depending on the concentration or the dose that is given. And then the resistance to them shows up in altering the penicillin binding proteins or hydrolysis of the beta-lactam ring. That's when the organism produces what we call the beta-lactamases, or changes its own penicillin binding protein so that the antimicrobial can no longer recognize the particular protein and therefore cannot bind to it. So those are the two main mechanisms of resistance to beta-lactam antibiotics. I've already explained this. Like I said, I'm flipping through so that we'll go to the new and emerging. The adverse effects, I've talked about that. And they're still very well used, particularly when you're able to dose them and keep to the intervals. That's usually the challenge in dosing beta-lactam antimicrobials, because most of them are taken either 6 hourly, 8 hourly, and at best 12 hourly. So the usual problem is adherence. But if the patient is able to adhere, they're still effective against quite a number of organisms. And then you have the 5 generations of cephalosporin, the first, second, third, fourth, fifth. And just remember that the lower the generation, the more gram positive. The higher the generation, the more gram negative. So you have gram positive activity in the first generation, highest. Coming down to second generation is still there. Then the third generation is more of the negative than the positive. The fourth and fifth usually have combined positive and negative activity. Most of them are administered parenterally, but of course, you know, we have a couple of orals that are also very good. And the advantage is that when we have to do IV to per oral conversion, we're able to get something that is very, very effective to continue from the parenteral. They're also not distributed widely, okay? Most of them are excreted unchanged, except ceftriaxone and cefoperazone, which also has biliary excretion. And of course, remember that that comes in handy when you really need to dose a patient who has really, really compromised kidneys. So ceftriaxone will be a choice because the excretion is not only via the kidneys. But even in that, depending on how severely compromised the renal function is, you still have to renally adjust the dosage. All right, and then you can read all this. Okay, so we go to the carbapenems, which unfortunately in several quarters now are being used almost as commonly as the cephalosporins, which should not be. If you remember our WEC classification, they should be in the reserve and 1 or 2 in the watch, but mostly in the reserve. They have very broad spectrum, especially imipenem, doripenem, and meropenem. Ertapenem, I always say, is the exception. I use that to remember it. Ertapenem is the exception. That is E, because it has poor activity against Pseudomonas. But the advantage with ertapenem is that it's administered once a day. Please recall and always bear in mind that imipenem will have the ability to induce seizures. It has the highest propensity to induce seizures. So usually for patients who are already seizure prone or whose seizure thresholds have been reduced one way or the other, they have inflamed meninges or something up there, please, you do not use imipenem. And remember that unfortunately, once the meninges are inflamed, most of these drugs that ordinarily will not have crossed the blood-brain barrier will cross it very, very readily. So it's something you also need to bear in mind. Remember that you also have to give IDPaN with cilastatin to prevent nephrotoxicity. And they're all used for mixed aerobic and anaerobic infections and also intra-abdominal infections. But please, not as a drug of first choice. You would usually think about piperacillin, tazobactam before you come to this, all things being equal. Okay, meropenem is metabolized to an inactive metabolite and excreted in urine. You can see the half-life is very short, but in renal impairment, it is prolonged. Ertapenem can be given IM, it's 90% bioavailable after that, and the half-life is a little longer than meropenem. IMIpenem has a half-life of 60 minutes and is also excreted almost 70% unchanged. Doripenem has a longer half-life. Okay, about the same half-life, length of half-life with IMIpenem, 1 hour. All of them, their half-lives are increased in compromised renal function. The adverse effects are known. I've already said what they'll be used for. You have the monobactams, and let's go to the glycopeptides, of which essentially we use vancomycin in our own area. And it has activity against all things gram-positive, except, of course, the ones that are specifically vancomycin resistant, some Enterobacteriaceae and some Staph aureus. And then you have televancin, which is a newer molecule under the glycopeptides. Those ones are more effective against the multi-drug resistant Staphylococcus aureus. So it still inhibits biosynthesis of peptidoglycan. And the basic mechanism of its resistance is formation of receptors on those peptidoglycan walls that have reduced glycopeptide affinity. So they have high affinity for something else apart from the glycopeptide. So the binding is decreased. And of course, once the binding is decreased, the permeation of the cell wall is decreased, inhibition of cell wall synthesis is decreased, and so on and so forth. It's poorly absorbed, so vancomycin is always given IV, except in cases when you need to sterilize the gut or in C. difficile diarrhea, where it is given to achieve local action. It's very nephrotoxic, very ototoxic, so you be careful when you're combining with other, should not actually be combined with other nephrotoxic or ototoxic medications. All I would want to point out very clearly in the adverse effects, amongst every other thing, is what happens when you give vancomycin IV rapidly. You know, so it's something you need to watch and warn about. Please, vancomycin IV should always be given by slow IV, preferably in an infusion, not as an IV push, because IV push, no matter how, unless you're sitting by the physician, you know, sometimes he's thinking about the other 4 or 5, 6 patients that he still has to attend to. So it's better to hang it on a bag and then set the rates so that the patient does not have the hypotensive reaction. Then you have our good old with fluoroquinolones, which in our parlance has become pure water, unfortunately, and therefore also having a whole lot of resistance to them. Even levofloxacin, that was supposed to be some kind of a reserve drug, is now being used. You know, everybody has accepted the fact that they have a kind of murdered ciprofloxacin. So levofloxacin now provides a clear alternative. The adverse reactions are known. They cause seizures, hallucinations, insomnia, and please, the seizure causing or the propensity to cause seizures of fluoroquinolones should not ever be taken for granted. Most patients in my practice that I have seen who have reduced seizure thresholds usually succumb to seizures even after just a single dose of fluoroquinolone. So please, it's not just academic, it's actual, something that all of us need to, keep our eyes on. All right. I want to go to the aminoglycosides. Very toxic class has dose-related nephrotoxicity, ototoxicity, and in addition, it has neuromuscular blockade, but they're still very excellent, especially in synergizing with beta-lactam. So that's why you see a lot of maybe ceftriaxone with gentamicin and maybe ampicillin, ampicillin, what's that again?
Clavulanic.
Yes, thank you. Amoxicillin, clavulanic acid with gentamicin. You also see a lot of it in malnourished kids that have what we call SAM, that's a severe acute malnourishment, malnutrition. They use it, sparingly at a very low dose, all the same, but they are very good at synergy, but they are very toxic. And the toxicity is dose related. So it's something you have to, thank goodness that I think the bolus formulation has been pulled from the market. I don't see it anymore, unless maybe somewhere else. That's the 280 milligram, which was causing a whole lot of damage. I think it was pulled several years ago. So I think what you get about here is about the 80 milligram, usually. If you still have the 280, please, there's nothing I can think of that would qualify a patient to be on more than that 80 milligram for a dose. So please, let's be mindful. And then could be once daily or extended interval dosing, or we could do the 3 times a day, but actually the extended as infusion would have been ideal. And it has a poor distribution into many tissues. And I've mentioned that it's best in synergy, not necessarily for monotherapy, but they're used for serious infections and they're bactericidal. Okay, the mechanisms of resistance are simple for you to understand. And I'm just trying to keep to the time because I know we lost a lot of time. If at the end we have time, you can bring me back to anything you want me to come back to. So we go to the macrolides and the ketolides, which were usually not so widely used until the COVID. And since the COVID, the use is yet to drop. And so I fear that we're looking at, in a couple of years, very high rates of macrolide and ketolide resistance. And they are our go-to class of drugs against the atypical organisms, and they are excellent against them. Very, very good against them. And they are good in the sense that children can use it, adults can use it. But the problem they have is that they prolong QT interval. Please remember that azithromycin has a very long half-life. And so usually a 3 to 5 day course is usually adequate. I've seen studies that have shown that there was no difference, absolutely no difference in outcome between a 3-day course, a 5-day course, and a 7-day course. So if there's no difference, why are we doing the 5-day? Why are we doing the 7-day? All right, so it's used for upper respiratory tract infections, lower respiratory tract infections, atypical mycobacterium infections, and so on and so forth, and Helicobacter pylori eradication. Then you have the tetracycline and the glycylcyclines, doxycycline, minocycline, tigecycline. Broad spectrum use, but there's resistance, particularly to the younger tetracyclines. But tigecycline is a bit better, in activity and less resistance. Very good against the enterococci, the multi-drug resistant staph, and so on and so forth, and very acceptable activity against the gram-negative rods and also the anaerobes, including our troublesome Pseudomonas. Remember that they don't do well with cations. And what I know with the tetracyclines is that sometimes they cause GI irritation, vomiting, and diarrhea that is very clinically significant, can actually send a patient back to the hospital. So it's something, just the same way we ask patients, do you react or do you have an allergy to penicillin? You need to ask them if they've taken tetracyclines before, if you're about to give it to them. And you need to ask how well taken it was. Sometimes you could minimize the GI irritation by taking with food, but also realize that the absorption is also affected. The oxazolidinediones, linezolid, very good, broad gram-positive activity. Unfortunately, because we're having high rates of vancomycin resistance in a lot of places now, we're beginning to fall back on linezolid. Those of you who have ever used it know it's expensive and it's toxic and it's challenging to actually set up and all that. So that's where we find ourselves because of antimicrobial resistance, but it has very poor activity against all gram-negative and anaerobes. And the major issue with linezolid is bone marrow suppression. You have the lincosamides as clindamycin and the brother. Very good against gram-positive, particularly like it in anaerobes. And it's very, very effective in skin and soft tissue infections. My only challenge with this is that you have a lot of, this is just my own, will I say, discovery, which I haven't subjected to scientific evaluation. But we have a lot of substandard versions of it. And so it's challenging. So when you get the lincosamides that you expect to actually be effective, you've done your culture and the organism is sensitive, then you get a particular brand of it and it's not really working as well. So it's something that we as pharmacists, it would be good for you to find out which brands actually contain the most active ingredients and all that, are able to release the best, and then kind of stick to those brands so that we don't make a mess of what we have. And then I have this, I call it my wonder drug. I do not yet understand how come till now we don't have metronidazole resistance that is significant. I just take it that is God's gift to all of us in this world and in Nigeria especially, because it's very cheap. Even with all the increase in drug prices, I think it's still about 5 naira or thereabouts for a tablet, which is not what you can say about most other antimicrobials. And it's very, has very good activity against gram-negative and gram-positive anaerobes and a lot of the GI organisms. The problem, of course, is the nausea, diarrhea, vomiting, sometimes pancreatitis, and of course, you have to advise your patients not to take alcohol while on metronidazole and tinidazole. So it has excellent bioavailability, almost 100%. The IV to PO bioavailability is almost equal. And then we use it for abdominal anaerobic bacterial infections, vaginal trichomoniasis, and protozoal GI infections. Also for Clostridium difficile infections and Helicobacter pylori eradication. However, because of its effect on the GI flora, it can actually cause, set up the patients for colonization with what we call the nasty organisms like the vancomycin resistant enterococci. The nitrofurans are still excellent activity against E. coli, and because a lot of us have left them behind, you find that sometimes when you do them on cultures, you see there's quite very frank activity. Okay, so, but the utility is limited to lower UTIs, not for urosepsis and all that. So let's look at the emerging antimicrobials. We have 8 new antimicrobial active ingredients, including one for the treatment of tuberculosis, approved since 2017. We had the pipeline up until I think 1997, 1999, we're having, rapid discoveries of antibacterials. And then as at 2019, it dried up till almost 2010 before we started having. And even at that, most of what we have are just modifications of what we had before. There are very few that are new molecules. All right. And the target of the new or emerging antimicrobials are those multi-drug resistant organisms, like the carbapenem resistant Enterobacteriaceae and all its other brothers. And then the ESBL, the extended spectrum beta-lactamase producing Enterobacteriaceae. Those ones are also very, very challenging to manage. We have more than 60 products that are in clinical development. And of course, you know that many of them will not reach the final line. And most of them consist of combinations of new beta-lactams. Remember I told you, modifications. And then new beta-lactam lactamase inhibitors. And then you have the ones in clinical development targeting at least one of the critical gram-negative pathogens. Remember at the beginning of this presentation, I mentioned that the gram-negative organisms are more challenging to battle. Forget the fact that we make a lot of noise about Staphylococcus aureus. It deserves a lot of noise actually, but when you meet the multi-drug resistant gram-negative infections, it's not something you want to handle. All right, so antimicrobials are still unable to treat the CRAs, and all that. Now, there's a polypeptide antibiotic called mirepavadine that was being tried, the IV formulation. It showed promise, but it had serious adverse effects, particularly nephrotoxicity. So it was discontinued, and they're working on seeing if they can get the inhalational form, hoping that the inhalation form will bypass the kidneys. So there will be no need, or the risk for nephrotoxicity will be low. And the advantage of this mirepavadine is that it does not exhibit cross-resistance with the same class of antimicrobials, which now is a problem with most antimicrobials, having the cross-resistance. So resistance to one becomes resistance to many, or resistance to everyone. So mirepavadine is different, but it's still under, will I say, trial. Then we have the zoliflodacin and gepotidacin that are in phase 3. You have the lefamulin, and then a combination of a beta-lactamase inhibitor, relebactam, with imipenem and cilastatin. And I want you to imagine the effects on the kidneys, But the problem we have is that some of the newer agents will be a lot more toxic and definitely a lot more expensive. And so that's why we would always talk about antimicrobial stewardship, so that we can preserve the ones we have for as long as we can. Then we have the cefiderocol, which is a beta-lactam antibiotic, active against the 3 critical priority pathogens that the FDA lists for complicated urinary tract infections. And recall that I said that most of the so-called emerging antimicrobials are modifications of the beta-lactams, modifications of the fluoroquinolones, modifications of the macrolides, and so on and so forth. So we have the zoliflodacin for the treatment of multi-drug resistance gonorrhea. And The good thing about it is its unique mechanism of action. It inhibits type 2 bacterial topoisomerase by binding to a different site than that which binds the fluoroquinolone. So that means that the organisms that exhibit fluoroquinolone resistance will not be able to exhibit the same resistance to zoliflodacin. So it's effective against multi-drug resistant gonorrhea and Neisseria gonorrhea, and some gram-positive and gram-negative bacteria. Please recall that I personally in this country, I haven't seen most of these drugs, but it's good we know that they exist, and maybe sometime when somebody who can afford it and is in need of it, then you know where to go to. Then you have the ridinilazole and bezlotoximab for C. difficile infection. Usually the most common health-related nosocomial infection in the world. I keep asking myself, and I don't know if anybody here has the answer, how come most of the data we get about C. difficile is from outside this country? Is it that Nigerians, for instance, or Africans in general, do not really have or suffer from C. difficile diarrhea? Or are we underreporting? Or is there something else we're doing to manage it? I don't know. But I know that outside this country, the C. difficile is a very important condition. They keep track of the, what would I call it? How much of it, what's the word? I can't remember the word now, but they keep track of how many patients come down with it, and is used as
prevalence. The prevalence.
Sorry? Yeah, prevalence. Thank you. So it's used as an indicator of how well a health system is managing their antimicrobial use and their infection control. So I don't know how it is in our hospitals down here. Your guess is as good as mine. But it's a major problem because the drugs that we have that are being used for it, they are now, the organisms are becoming resistant to them. So you have the fidaxomicin, which is used in C. difficile diarrhea, and the advantage is, unlike others, you have minimal risk of damage to the normal microflora, but it's very expensive. Then we have the tigecycline that we've already had. You have the teicoplanin that sometimes you come across, rifaximin and bacitracin. You remember bacitracin? I don't know about you. In Nigeria, the last I know about bacitracin is that we use it as we get the bacitracin powder, if anybody can remember. We use it for treating wounds in those days. Because of antimicrobial resistance, we're now converting many of those things to see how well they could be used orally or parenterally. So the problem with the C. difficile is that in spite of all the so-called emerging therapies, reoccurrence still comes up. And those recurrent infections are usually very difficult to treat. You have the ridinilazole, which is also used for C. difficile, bactericidal, and inhibits the cell division of the bacterium, binding to the minor groove of DRA. It's orally administered and poorly absorbed, so it can act locally in the GIT. It has a limited systemic absorption, which is good. It has reduced action against the gut microflora, which is also good. So it's almost an ideal drug for the treatment of Clostridium difficile diarrhea. Then you have the monoclonal antibody, bezlotoxumab, is approved for prevention of recurrent CDI in adults. I don't need to tell you, as pharmacists, what the monoclonal antibodies look like and what their effects on the body systems are. So you can imagine with me what would happen with these patients who now need to take it to prevent recurrence. It's given as a single dose in combination with an antibiotic treatment against C. difficile. So it's not effective in monotherapy. Use the ordinary or the well-recognized therapy for C. difficile, and then you add on bezotoxumab to prevent recurrence. It was approved in 2016. Then you have a new tetracycline derivative, relatively new. It's a fully synthetic fluorocycline and belonging to the tetracycline class. And together with the relebactam we had mentioned before, relebactam is a beta-lactamase inhibitor, a new beta-lactamase inhibitor, which is combined with imipenem cilastatin to reduce resistance to it and make it more effective. But eravacycline was specifically developed to overcome acquired resistance that is experienced with traditional tetracyclines. Sorry, I skipped over it. If you go back to my slides, you'll find out we talked about how tetracyclines, how resistance to tetracyclines occur, which is, first one is the acquisition of genes encoding the efflux pump. So they cause, how do I put this? The tetracyclines act by binding to the ribosomes. So they need to be, excuse me, they need to be absorbed into the cells. And so the organisms that show resistance to tetracyclines develop efflux pumps. So as the tetracycline is being pulled in, those efflux pumps are pumping the tetracycline out, preventing them from accumulating in enough concentration to actually carry out their effects. So that is one primary way of resistance. And then also the presence of ribosomal protection proteins that prevent the tetracyclines from actually attacking the ribosomal proteins that they intend to. But the good thing about the eravacycline is that the efflux pumps do not recognize it. They do not recognize it as a tetracycline. So it does not, it doesn't get pumped out like other tetracyclines. And it's been approved in intravenous forms. But remember, I said they're all very, very expensive. Then you have another fourth generation fluoroquinolone, delafloxacin. The difference with this is that it is, in terms of stability, in terms of in vitro activity, corresponding to in vivo, it's better than the other classical fluoroquinolones. And it's used for treatment of acute bacterial skin and skin structure infections, usually caused by multi-drug resistance strains. It has an increased activity in acidic medium, unlike the other fluoroquinolones. And it's a weak acid, and at neutral pH, it's an anion. So you find out that it is effective in acidic medium. Another advantage of this, of delafloxacin, is that although the peripheral neuropathy that is reported with all the fluoroquinolone class is also found with it, it does not cause QT interval elongation. So that means for patients with heart disorders, you may be able to use delafloxacin. It also found not to be photosensitive, doesn't cause photosensitivity. And then remember that I said most of these drugs have been in circulation for just 5 to 6 years. They are expensive. That means that you don't get to see so many patients using it. So the post-marketing surveillance on all these things are very poor. It's only when the use becomes very wide, then you can really say for sure that this is safer than the other or not. You know, what we call about the law of large numbers. Then you have the new beta-lactams, mostly combinations of new beta-lactamase inhibitors and all that. You have the taniborbactam with cefepime. Remember, cefepime is a 5th generation. And then you also have this one, I think is already in our space, ceftazidime and afibactam, which was approved for treatment of complicated UTI, and quite effective, but of course more expensive than the plain. Then you also have meropenem with vaborbactam, also for complicated UTI, for complicated intra-abdominal infections, for hospital-acquired pneumonia, for ventilator-assisted pneumonia. And this vaborbactam, unlike most of the younger beta-lactamase inhibitors, inhibits many class A and B lactamases and carbapenemases. Most of the problem we've had with resistance to carbapenem are the organisms that produce carbapenemases, and most of the beta-lactamase inhibitors have not been able to tackle them, but vaborbactam does. So what we will call the emerging antimicrobial is that they now add vaborbactam to the beta-lactam antibiotic, and then it's called a new molecule in quotes. And I say in quotes because it really isn't. It's just a modification of it. But the combinations have shown very good activity and very high tolerability. And the adverse effects are also not very significant. Then you have the relebactam, which is also similar to avibactam, and then has been added to imipenem cilastatin and has been shown to be very effective against organisms that ordinarily are resistant to imipenem. Then you have a new aminoglycoside, plazomycin, very effective against gram-negative infections, for which gentamicin has been rendered ineffective. And very interestingly, plazomycin is able to block the aminoglycoside modifying enzymes. If you go back to the notes I have on aminoglycoside and how the resistance occurs, you will see that one of it is production of enzymes. So the plazomycin is able to block those enzymes that are produced by the organism. So resistance to it for now is very, very low. And it differs structurally a bit from the other aminoglycosides. So it has been subjected to 2 clinical trials, and in both studies it has been well tolerated, but we have noted reversible ototoxicity. It was also approved in 2018 in the United States. Then you have a new cephalosporin, cefiderocol, belonging to the later generations, and is specifically for multi-drug resistant bacterial strains. It's effective against complicated UTI caused by gram-negative community-acquired bacterial pneumonia, ventilator-assisted or associated bacterial pneumonia. Structurally, it's similar to cefepime, which is a 4th generation cephalosporin. We call them zwitterions. They are not anions, they're not cations. They are zwitterions, and it allows them, they are mostly anionic states anyway, allows them to penetrate better into both the gram-positive and the gram-negative organisms. And then because it's been added, it has an added C7 side chain, carboxylic acid in the C7 side chain, which is common. The C7 side chain is common to all cephalosporins. It improves the permeability in the outer membrane. So it's able to get across the microbial membranes better, and is also stable towards hydrolysis by the enzymes. Very, very effective against almost all the isolated strains of those our troublesome organisms, Enterobacteriaceae, Acidobacter baumannii, Pseudomonas aeruginosa, and all that. So it's been approved as an injectable drug, a parenteral drug. And it has also been approved for treatment of adult patients with complicated UTI, including renal infections caused by sensitive gram-negative organisms, which have limited treatment options or no alternative at all. And in 2020, the indications were upgraded to allow treatment for hospital-acquired bronchial pneumonia and ventilator-associated, I don't know why I keep saying assisted, ventilator-associated bronchial pneumonia caused by A. baumannii, E. coli, and all that. And there was a pilot study on patients with chronic UTI, and they showed that up to 70% had a resolution of symptoms and eradication of bacteria 7 days after treatment, as compared to 54% of those who received an alternative. And the beautiful thing, the thing that sets out this drug above others, is that it's been able to overcome all the 3 mechanisms of development of resistance to beta-lactam. So it does not respond to bacterial beta-lactamases and all that, including the efflux pumps and the modifications of the purines. None of them affect it. So for now, it is immune to any resistance, any plan, any ability of the organisms to become resistant to it. For now, there's no such thing.
Sorry, ma. Hello, ma.
Yes.
Yeah, we have about 5 minutes more, ma.
Okay, I'm almost done.
Thank you, ma.
You have the, this one is for MDR tuberculosis, pretomanid, and it's also acts in a different way from other anti-TB medications. It inhibits the ATPase proton pump that supplies ATP to the microbacteria, but it's been shown to be effective against multi-drug resistant TB. All right. It also blocks cellular respiratory processes in an anaerobic environment. So it kills the M tuberculosis through different ways. So it is effective in both the latent one and the replication, the one that are actively dividing, which is something that is special about it. And it's also active in both aerobic and anaerobic. So whether it is, what do you call it? Even if it is the normal TB or the miliary, you can use pretomanid. You also have the delammanid, similar to pretomanid, and they are both effective against the most aggressive and severe forms of tuberculosis that are not able to be treated by traditional drugs. Please note that out of all these things I've said, only 2 represent a new chemical scaffold, a new chemical entity. Most of the others, like I had mentioned, are derivatives, modifications. Take a side chain here, put it there. Break a bond here, put it the other way. But there's significant progress in research, especially for antimicrobials that are going to be effective against gram-negative bacteria, which is a major challenge. It's also important that the new antibiotics that we are looking to have do not have cross-resistance with other existing compounds, because if they do, they're already dead on arrival. So the search for new and emerging antibacterial drugs are resulting from modifications of the traditional ones, keeping in mind their ability or the knowledge of how cross-resistance is developed. Of course, finding the new ones are very difficult. And because it takes a very, very long time to clinically develop a new molecule, we expect several new antibiotics to be approved in the next couple of years, while we expect that a lot of them that have entered the race may not get to the finish line. And that is the world of drug development. So thank you for your time and attention. And I hope I've been able to communicate at least somewhat in this short time. Thank you.
Thank you very much, Ma, for the loaded presentation. Very interesting and captivating. Save for time, we'll have stayed all the night to listen to you. Thank you so much, Ma. God bless you.
Thank you.
Dr. Onodugo has been able to unpack a lot about antimicrobials, the traditional ones that we're used to and the emerging ones. She has been able to identify the importance of identifying the type of organism. Is it gram-negative? Is it gram-positive? So that we can manage the infections better. She has talked about the time-dependent one that will determine the dosage interval, or the concentration-dependent one that would determine how large the strength of the drug we're using should be. She has talked about different classes of the traditional ones and the emerging ones. And she has emphasized severally that the emerging ones, most of them are modifications of the traditional ones. So won't we preserve what we have with us? Because tonight she has told us that a lot of the new ones, though might be very effective, but they are very expensive. Considering our setting here, they will be very scarce, probably not accessible. And post-marketing surveillance will be very poor, especially among us here, the Blacks. So tonight, I won't take much of our time. I would like to take some few questions, comments, contributions. So if you would like to contribute or ask questions, please raise your hand electronically so that we can identify you and call you. Thank you. Comments, contributions, questions. So she talked about the different antimicrobials. There are different classes. There are probably adverse drug reactions. Okay, so right now I have Dr. Hime Eshet. Please, can you unmute and speak?
I'm here. Thank you very much, the presenter, for a very insightful lecture. Yeah, I really appreciate the fact that you took time to tell us where we were in terms of antibiotic availability and where we are today. Then my question is, you said most of the new antibiotics or emerging antibiotics are basically modifications, structural modifications from the existing ones. So my question is, do they really offer any therapeutic advantage over the other ones that are already here in terms of resistance? If it's going to be the same thing, then why are we bothering ourselves with them? Because if they are going to be, they'll be resistant to them already, then I don't think there's need for us to have this new one that are basically structural modifications from the existing ones.
Thank you, Dr. Eshet.
That's my question.
All right.
Okay, do you want me to answer it or you want me to take all the questions?
Yes, ma'am.
Okay, so yeah, even though there are modifications, Dr. Eshet, thank you for your question. There are certain therapeutic advantages. If you listen to me well, some of them were actually, like the last one we talked about, even though it's a structural modification, that modification allowed it to be immune to the 3 known mechanisms of resistance to other members of its class. So, but remember, I'll keep saying for now, because you know that resistance is a natural phenomenon. And all bacteria are made to develop resistance to antimicrobials. One day. All we're pushing for is to push that day very, very far. If we use antimicrobials well, if we use them the proper way, we don't misuse them, we don't abuse them, yes, we can push that day far. So yes, there are advantages. Some of them are even the modifications that made them less toxic, while some are marginally more toxic. So, and remember that I mentioned that all these emerging antimicrobials are for multi-drug resistant infection. So it's either you use those new ones or you watch the patient fade away before your eyes. So yes, there are advantages. And we're hoping that the advantages will prevail for a long time. I saw somebody on the chat box who said, the structures, do they affect the resistance? I think I've answered it. Most of the structure, because what they are doing now is they are modifying the structure, looking not just at the mechanism of action, but at the mechanism of resistance and the mechanism of cross resistance, because the problem is the resistance. So what they're doing is modifying to make the new molecule or the modified molecule not easily resistant, that the organisms can no longer raise the resistance to them as easily as the other traditional ones. So yes, there are advantages to them, but they're very expensive. And yours truly, I haven't seen them in Nigeria. I don't know who has seen, you know, and I imagine they will be very, very expensive anyway.
Thank you, ma. So, thank you. Thank you for answering the one on the chat too. So do we have more comments? Comments, contributions, and questions? I'd like to say thank you again, ma, for making.
Professor
okay, Dr.
Jordan.
Jordan. Okay. Dr. Jordan, please take.
Thank you. Just a bit curious to find out if these emerging antibiotics, if they have made it into the aware classification yet, and if yes, where they fall in that aware classification. Just wondering, are they that new? Are they so new that they have not yet made it, or they have been included in the aware classification? And if yes, if they fall within the 3 classes, or they are all on reserve or something? Just curious, just being curious. Thank you.
Okay, I may not be able to answer that very fully. However, I can imagine that anyone that has made it should be in the reserve. Because like I mentioned, all of them are for multi-drug resistant organisms, infections. So they should definitely not fall under the access. They probably will not fall under watch at all. So I believe that anyone that makes it to that aware classification should find itself in the reserve. But because I didn't check that, I cannot answer that very, very emphatically.
No problem.
Thank you.
Thanks. Thanks for your insight.
Okay. Charles Akishete.
Yeah, thank you very much. Good evening, everyone. Thank you, lecturer. That was very decisive and deep. I want to also look at it from another angle. In the School of Science, there is the angle of business. If you have a product in your market, in the market space, and that product does not have another innovation that surpasses the one that you have been, the product you have been marketing, it gets to a stage where that product's life cycle will go off. Every product has a life cycle. And after that time, it disappears from the market space. So to be in business, both as a scientist and as a company and researchers, you also must develop new molecules and new products. Just the same way we have IT, your phones, Pro Max 1, Pro Max 2, S4, S10, give it different name, change the RAM, change the camera, and it becomes appealing to people. So you must, as scientists, we must be innovative. And just like she said, definitely there is a timeline for all products. I remember very well as a young marketer then when we were marketing Tarivit, ofloxacin. It was as if ofloxacin will not leave the market space. Yes, it's still there, but it's not as commonly used as it used to be those days. And I remember when Cipro Flosacin came, just a little modification to the structure, it became a bank. And today we have Cipro plus tinidazole, Cipro plus onidazole. So we must keep on innovating. And I think that is the hallmark of science. Thank you.
Thank you very much, Mama Kinshete.
Sorry, please, could I just say something because of the examples he used at the end?
Okay, ma'am.
As antimicrobial stewards, we're actually very, very uncomfortable with those fixed dose combinations. We believe that they are part of our problem. So you find a lot of people using ciprofloxacin and tinidazole when they may have only used tinidazole and been okay, or may have only used ciprofloxacin. So those fixed doses combinations are also troublesome, in terms of antimicrobial stewardship. We really we're not very comfortable with them. I just needed to mention that in passing.
Okay, thank you, ma'am, for that. We really need to be careful about combining different antimicrobials because of resistance. Thank you, everyone, for this evening. In the absence of any more comments, contributions, I'd like to say a very big thank you for Dr. Mrs. Chineye Onodugu for this insightful presentation. Thank you for your time. Thank you for your efforts. Thank you for presenting it in an exciting way. We're grateful. Thank you, ma. And I would like to say thank you to the audience. Thank you for being attentive and participative. We appreciate your time this evening. We are not taking it for granted. Thank you. And I don't know if Dr. Joda would like to give closing remark before we take the closing prayer. Dr. Joda.
Yes, ma. Thank you, moderator, for the responsibility of giving the vote of thanks. I would just like, was it vote of thanks I'm asked to give or closing prayer?
Closing remarks.
Okay, sorry, closing remarks. Okay, so I was just writing in the platform, actually. That's why it took me a second to come and unmute myself, that it was really quite an enriching presentation. Thank you, Dr. Nodugo, for effortlessly, going through these antibiotics and anti-infectives and drawing our attention to important things about each of these things that a lot of times we have taken for granted, but which we should not take for granted because it helps us to be able to do our jobs better, and for those of us teaching, also to teach better. So thank you very much for not only the presentation, but the richness of the notes that you are dropping for us. That will be a very good resource for all of us going forward. So I'm happy that I was here, and I'm glad to say that I believe we're over 70 people in this presentation at some point, and that's a very well attended event by the infectious diseases pharmacist group. So thank you very much, everybody, for making this such a great outing. Thank you, Dr. Nadugo, and thank you, Dr. Issa, for being the engine behind the engine. We appreciate everybody. Thank you so much, everyone.
Thank you, ma. Dr. Eshiet.
Thank you.
Please, can you give us the closing prayer?
Okay. Can we bow our hearts in prayer? In Jesus' name.
Amen.
Father, we give you all the glory, all the honor. We thank you for today, and we thank you for what we have learned today, and we thank you for guiding us through this journey of antibiotic stewardship. Father, we ask for grace for us to be stewards indeed, and we ask for grace for us to impart what we have learned today in our various areas of practice. And we ask for your blessings and your guidance for our association. And we ask for you to bless and keep us. And as we go to our beds, may we sleep peacefully and wake up stronger and healthier. In Jesus' name we are praying. Amen.
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 Thank you, everyone.
Amen. Thank you very much.
Until the
Amen.
next
Thank
evening.
you. Good night,
Amen. Thank
everyone.
you very
Good
much.
Bye,
night.
everybody.
Bye-bye.
Bye, everybody. Bye.
Thank
Bye.
you.
Bye, everybody.
Good night.
Bye, everybody.
Bye.
Night.